So yeah, I'm actually really excited today to be able to host Natalie Grefenstette, who is currently a postdoctoral researcher at the Santa Fe Institute, who's working on the Agnostic Biosignatures project, which is a multi-institutional NASA project looking at many different facets of the agnostic biosignatures. Um, so Natalie got her degree from University College London, her bachelor's degree in University College London. And I think probably like a lot of people on this call said that she was, originally in sort of from a early age, just interested in these questions of what is life? How did it start? What was the environmental constraints? So she went to University College London, got her degree in biochemistry, and then stayed there to do PhD work in prebiotic chemistry. So I actually met Natalie through the Astrobiology Primer project, where she is the co-lead editor for two different chapters, both on biosignatures. The first being biosignatures, the chapter where they go into the different techniques and types of biosignatures. And then she's also the lead editor for the chapter on alternative life, which I think fits very nicely into this agnostic biosignature picture. So with that, I will let you take it away, Natalie.
Hi, everyone, and thanks for having me. Thanks for the introduction. I just want to check you can all hear me okay? Nod
All
from
good.
anyone or anything? Yeah, all good. And if you do see anything weird, like notes that aren't supposed to be there or slides that aren't advancing, just let me know. Yeah, so as Micah said, I'm currently a postdoc at the Santa Fe Institute, and as I was doing prebiotic chemistry in my PhD, I'm now actually doing more theoretical astrobiology at the Santa Fe Institute and doing more computational work and modeling and things like that. So it's been an interesting shift for me. And so today I want to tell you a bit about the project I'm involved in, the Laboratory for Agnostic Biosignatures, and how we're thinking about detecting life as we don't know it. So this talk will be less focused on specific results and more trying to give you a general picture of what that means and what do we mean when we talk about detecting life as we don't know it and how we go about that. Like, how do you even look for something when you don't know what you're looking for? So the questions in astrobiology, I think that we're all interested in are these questions here that hopefully you see this slide. I'm going to assume that that's a yes. So what is life? How can we recognize it? And how does it emerge? So these are questions among others in the astrobiology community that we're interested in and questions that I'm particularly interested in. And today I'm going to focus on the second question, how can we recognize it? So as we all know, one of NASA's missions is to search for life in the solar system and beyond. But the question to ask is, what are we looking for exactly? So traditional astrobiology has focused on looking for signatures that are closely associated with life on Earth, as that's the only example that we have. But there's a move now towards generalizing our approach to search for life in the general sense and to be more inclusive to potentially very different forms of life. So here's a little diagram to help illustrate this idea. So here you can see the oval shape, which represents the space of signatures that life on Earth expresses. So we call these Terran biosignatures. So this includes some very specific signatures, such as steranes and nucleic acids, which are used to detect microorganisms in Terran environments or in the geological record. So some subsection of Terran biosignatures, on the other hand, are more general. So these are more general biosignatures that are potentially universal features of life that would be true for any form of life in the universe. So we call these universal biosignatures. And at this point, you might realize we don't actually know which signatures of life are universal just yet. And if we did, our job would be done and it would be great. But so how big or small this circle is in terms of the proportion of signatures of Terran life that is universal is currently unknown. So in this diagram, as you move away from the center of the universal circle, the signatures become more specific to that type of life and its evolutionary history on whatever planetary body it evolved on. So this is based on the idea that if life evolved in a different environment with a different history and a different geochemistry, life could look completely different. And so the specifics of the biochemistry of that life and the signatures that it then produces would be very specific to that life form and the planetary body and would be currently unrecognizable to us. So in the case of Mars as an example, you can see one of the petals now is labeled as potential Martian biosignatures. So we know that Mars, even though in many ways it's very similar to Earth or was in its past, it's gone through a different and independent geochemical evolution from Earth, which would have likely driven potential life forms to look different than ours, and so biosignature to look different from Terran biosignatures. So this is especially true if Earth and Mars did not share a common biological ancestry. Therefore, if we focus on search efforts on universal biosignatures, the center of the circle, we have a higher chance of capturing signatures of life on Mars because we're not biasing ourselves towards Terran biosignatures. And I just want to point out here that it's important to acknowledge that while we think that these different petals would experience different geochemical evolutions and different life forms would experience completely different histories, there's no existing knowledge or evidence that life elsewhere would be different or that it would be the same. So this is just trying to kind of make it more general just in case it is different to life here on Earth. So the best that we can do is relax our assumptions about the requirements and features of life so that we are broadening our efforts to encompass more universal features that may be present in other life forms. So as I said earlier, we don't know which signatures of life are universal yet. So this is where the idea of agnostic biosignature comes in. So agnostic biosignatures are signs of life that do not presuppose an underlying biochemical system. And so it's agnostic, we don't know, to the biochemistry of the life form and the planetary conditions that we're exploring. So these agnostic biosignatures are trying to get closer to the concept of universal signatures of life by abstracting what we know about life and trying to find more general ways of looking at life and its signatures. So this is in the hopes that if we did come across life form that is different than our own on a mission, we'd be able to recognize it as life so that we don't end up in a false negative situation where a planet or an environment actually has a life form, but we're so focused on a certain known signature of Terran life, like nucleic acids or steranes, that we actually miss it. That would be such a shame to spend so much time and money and actually miss this opportunity. And importantly, I also want to point out that these proposed agnostic biosignatures would also be able to detect life as we know it. So it's not trying to have one or the other. It would just be more general and encompass Terran life detection. It would just be detecting it in a different way. And so I'll go into some examples later to give you a better idea. So I want to dive a little bit further on what we can do as a community to enhance our search strategy so that we can enhance our chances of recognizing life if it were different than Terran life and if we were looking for it in a mission, for example. So we know that within this proposed Terran biosignature oval here, there is all of our knowledge of what life looks like and the signatures it expresses are that we currently recognize, so the life that we know here on Earth. So that's this yellow oval represented here, what we currently know. So some of these signatures could already be universal, at least inclusive of some other forms of life. And some of this knowledge of what we know about life and its signatures currently overlaps with our ability to detect these expressions, thankfully, so that we can actually start looking for things, which is represented by this blue oval. So the area that overlaps between the yellow and the blue on the left shows all the detectable recognized Terran biosignatures. So what we know of traditionally as biosignatures, like specific molecules, homochirality, or biosignature gases. In the agnostic biosignature community, we propose that our search efforts should focus on where our detection capabilities, the blue, overlaps with our knowledge on universal biosignatures. So that's this little triangle on the right, or at least try to get as closely to that as possible through the use of agnostic biosignatures. So ideally, these newly proposed agnostic biosignatures would aim to include all possible life forms. But as we don't know exactly what that means, as I said earlier, getting as close to that as possible by generalizing what we know about our life is already an improvement and increases our chances of detecting life. So in order to do this, we can start by trying to broaden our knowledge base in order to increase the size of that yellow oval in the dark gray area on the right-hand side. So how can we do that? So this can be done by two different approaches. First, we'll look at the top-down approach. So this approach focuses on identifying key differences between life and non-life. So these could be chemical or physical differences that you can see in like samples or something. So you can examine existing data and determine the key chemical and physical differences between living and non-living samples, or samples that came from life and non-living samples. And when I say non-life here, I mean completely abiotic, not things that used to be alive, because that would just be still part of the umbrella of life when we talk about it in an astrobiological context. And then test these differences and these findings against a reference sample set that spanned the chemical space between biotic and abiotic to kind of understand this whole spectrum of life. So most of the examples I will show you later in a minute are based on this top-down approach of trying to just identify the differences first. The other approach is the bottom-up approach, which focuses on trying to identify universal principles of life and how those would impact signatures at the chemical or physical level. So this can be done through research and development, including modeling, theoretical, and experimental studies on universal principles of life, biophysical laws, complexity science, as well as research on the origins and emergence of life. So these studies would provide us with a deeper understanding of what life is fundamentally, as well as what the possible chemical and physical space that life can occupy are, and how we can use that knowledge to enhance our detection capabilities. So the second part I'll talk about a little bit later, and that's kind of what I work on a lot at the Santa Fe Institute. And ultimately, these two approaches should be used together in conjunction so that we can increase our understanding of how to find life, whatever it looks like. So I said earlier I'd give you some examples and recent advancements in the field. So that's what I'll show you right now. So first I'll start with how we could use an instrument that's traditionally used in astrobiology to look for agnostic biosignatures. So mass spectrometers are a great example of a high heritage instrument. So that's an instrument that's been used already in missions and over again. So it's been used since Viking. It's on board the Curiosity rover. It's going to fly on ExoMars and Dragonfly in the next several years. And so we know that traditionally mass spectrometers are used to detect Terran biosignatures, such as complex molecules that could be present in samples such as molecular fossils. So these compounds, like the ones here, are fatty acids and sterines. They're quite robust Terran biosignatures because their presence or distribution requires a suite of complex biosynthetic pathways to produce, and they're produced in high abundance. So they're tightly linked with Terran biology. But this contemporary interpretation is very specific to Terran biochemistry. So detecting one of these or a specific pattern of fatty acids is almost an unequivocal sign that we detected life when we're thinking about life in the Terran sense. But there are also alternative ways that we could interpret this data that we get back from mass spectrometers. So one way to do this would be to, instead of looking at the specifics of what the molecule is, instead we want to look at their intrinsic complexity, where molecules like short chain fatty acids may have a much lower complexity or information, and molecules like this sterane has a high complexity. And so the probability of it existing randomly just made through an abiotic process in abundance is very low. So this suggests the presence of life or lifelike machineries that are able of catalyzing specific reactions in order to make the same complex molecule in high abundance. So we don't necessarily need to know the specifics of the biochemical system needed to produce this in order to say that that molecule on the right-hand side has a higher chemical complexity, which is proposed as a proxy for information needed to produce that molecule. So current work at the University of Glasgow and NASA Goddard is being developed in order to use spaceflight-capable mass spectrometers to look for signatures of chemical complexity. So this is an example of how going from a Terran biosignature, we can kind of generalize the concept of, instead of just looking for specifics of a molecule, we're now looking for the complexity of that molecule as defined, like these teams are doing, and then how we can use current technology to look for that new biosignature. So another example is using nucleic acids as the Terran biosignature. So considering the central role of nucleic acids and other biopolymers in Terran biology, which are used for information storage, catalytic activity, and encapsulation, it's been proposed that other life forms may also rely on linear polymers for their biochemistry. So this is also a good example of a proposed general signature of life that might help include some other life forms, but not necessarily all life forms. We don't know if this is universal because it's unknown whether linear polymers are needed in general for life to exist. That's still being debated and researched upon. But some people do think that it is probable that other life forms also use some kind of linear biopolymer. And I'll come back to polymers a bit later in the talk as well. So current work that I'm involved in, along with colleagues at the Santa Fe Institute, the University of New Mexico, and NASA Goddard, is looking at how we can use high heritage instruments such as mass spectrometers that I already talked about to look for signatures of polymers that would include nucleic acids and, importantly, other unknown polymers that could be of interest as signatures of life. So this is even without knowing the identity of the polymers, how could we detect it using mass spectrometers? Another interesting example that I wanted to showcase is of a proposed agnostic biosignature that's being developed. That's actually based on repurposing a technology that's been developed for a specific pterin biosignature, in this case, nucleic acids. So this technology is a nucleic acid sequencer, like a nanopore, which can be used to detect long strands of DNA or RNA. But instead of using it just to look for nucleic acids, here it's proposed to be used in a very different way. So indeed, we know that these biopolymers are very specific to pterin life. Instead of looking for those ones, we could use this technology to probe for surface complexity or surface heterogeneity. So for example, the surface of cells, we know this, has a high level of complex expression because it has like all these different proteins on the surface and different types of fatty acids and everything. So when you compare that to a mineral that might only have a few different crystal faces on it, you end up with a much higher surface complexity on a cell. So the method being developed is called the Fingerprinting Method, which would take advantage of the fact that some short DNA sequences form small aptamers that can bind specifically to different types of molecules, and so they can bind to these surface expressions of cells or minerals, irrespective of the cell biochemistry. So then it can be used for any type of living system in theory. And then you can wash those off and study those aptamers using this nanopore technology. So this kind of technology developed by Georgetown is at the forefront of agnostic approaches that could be used for looking for example, extant life on Mars. So to come back to this little diagram here, we've been looking at some examples of how to expand that yellow area, so what we know, but still using high heritage or already developed technologies. So that's the overlap actually with the blue oval. So another tactic we could do is to also increase the types of instruments that are used for spaceflight in order to match newly proposed agnostic biosignatures and new ideas in this field, and so increasing that blue oval as well as the yellow. So some example of these are secondary ion mass spectrometer, SIMS, which is a technology that's not currently flight capable, but it's used on Earth to observe disproportionation of specific elements from the background. So this is a proposed agnostic biosignature because the idea is that irrespective of the elemental basis of a life form, this accumulation of elements could be indicative of the presence of life that is distinguishable from the background elemental proportions. And another one would be to look for detection of energy transfer, which is used by life on Earth often, by using electrodes and cyclic voltammetry. And these ideas are being developed and worked on at Penn State and Harvard. So now I want to give you a glimpse into some of the work that I've been working on, which is more on the bottom-up approach side of things. So the work that I'm developing is focused on this question: can we detect signatures of evolution? So evolution is a key feature of life that's talked about a lot in astrobiology, but understanding how we could detect that as a signature, considering we usually think of evolution in action as a long process over time, it's been very difficult to think about how to actually detect this. So we're trying to chip away at this question in the project that I've developed with collaborators. So first of all, how can we model evolution, and what do we mean by evolution? So in this project, we decided to break down evolution in two components that are often talked about as required and key components of evolution, which are selection and reproduction. And I'll come back to that in a little bit. So you've probably seen this type of timeline before. This one is heavily modified from Joyce to show how the emergence of life happens through time. And so you can see first an environment like a planet stabilizes, and then there's, prebiotic chemistry that emerges. Still, we don't exactly know how that happens. And then that leads to first evidence of life at some point. So I think you can look at this as a timeline, but also as a spectrum in a way. So it goes from fully abiotic, a pure physicochemical system, to fully biotic, a complex ecosystem, like the one that we have here on Earth now. And so this range of uncertainty and on the spectrum is sometimes called the grayness of life. And this would be true of life on other planets. And this means like we don't have a good definition of when this grayness ends and starts. So what really constitutes life, and when does it actually emerge along this timeline and along this spectrum? And arguably, one of the hardest places on the spectrum to make a biosignature detection would be at this boundary between pre or proto-biological systems and fully fledged life. And we don't know if there is a defined boundary and what that really looks like. And there are also a lot of interesting chemical systems that you can imagine in that gray zone, like prebiotic systems that could have led to the emergence of life here on Earth or other systems. So this is where kind of everything is happening. But we don't really know what that looks like. So in this project, we decided to try and understand a little bit more about this gray area and what kind of signatures of protobiology might exist in chemical systems and signatures of evolution in those protobiological systems. So in our model, we use polymers, like I talked about earlier, to study effects of evolution. So why polymers? As I mentioned before, historically, polymers have been a great interest to the astrobiology community and origin of life communities. Because they have a central role in terran biology and proteins, DNA, and sugar molecules, and because generating polymers, especially RNA and proteins, is a main theme in prebiotic chemistry, and understanding polymer interactions is prevalent in experimental and theoretical studies in both origin of life and also just biology in general. And looking for biopolymers like DNA or other biopolymers on other planets is also, as I talked about, a common biosignature that's discussed. But non-biotic polymers are also common in the universe. They can be made in different types of environments, so they're quite ubiquitous. So now if we're interested in the wider search for life, and we're not presupposing its biochemistry, and say we're in a life detection mission and we came across a population of polymers on another planet, what would be a dead giveaway that this population of polymers came from a biological system? So this really isn't a straightforward question. First of all, how do you even detect the polymers in the first place using technology we use in space missions? So that I alluded to earlier is a project that we're working on within lab to try and answer. Then how do you deconvolute the various polymers that are in there? Even on Earth, this could be quite difficult. So in this case, we built a simple computational model to study what the signatures we might be able to detect in populations of polymers are that would help us understand where along the spectrum this population came from, and what hurdles we should be aware of when we're in a life detection mission, for example, that might convolute the signal. So where could we easily get confused? What would be signs that are dead giveaways? Like all those kinds of questions. And as I said before, in our work, we're focused on this gray area that's on the spectrum, and a part of spectrums that mostly represents proto or pre-biological systems. So for this project, we built an out-of-equilibrium model that can form linear polymers that can grow over time. So polymers are attached, so this is all computational and it's a model, but I'll talk about it in a more physical sense. These polymers are attached from their side onto the side of this space, and the space could be seen as a sort of like a pore, a cell wall, a sort of interesting geochemical environment that has a surface basically that you can interact with. And the surface in this model is catalytic and encourages polymerization. So this is to kind of parallel work that's being done in Origin of Life, where catalytic activities from surfaces are being explored as a way to polymerize polymers. So in this model, monomers can flow in, react with other species or polymers or monomers that are already in this cell or pore, and then they can be flushed away. And so we're interested in using this very simple model to understand how different aspects of evolution can influence the system. So as a reminder, what I mean by evolution in this case is both selection and reproduction. So evolution, again, is often talked about as a key feature of life, and it's not just present in cellular life. Of course, it was also active beforehand in chemical systems. So I won't really go into the difference between chemical and biological evolution here, but it was definitely something that was already present before like first life really started. It's something that exists in every kind of chemical system. So how can we model this? We could break down evolution into the two key components I mentioned, selection and reproduction. So in our case, we mean fitness-based selection, so based on different attributes of the polymers, like their length or the different kinds of monomers that they're composed of. And by reproduction, we mean template-based reproduction, where you can make a copy of the polymer. So by switching these on and off, you get 4 different combinations. You can have, there's no templation, there's just random selection, which is a more abiotic kind. Then you can have no templation, but you do have fitness-based selection, or you could have templation with no fitness-based selection, or you could have the whole thing with templation and fitness-based selection present. So you can kind of see these 4 different combinations or 4 dynamics as along this kind of spectrum from more abiotic to more biotic. So these 4 dynamics help us model the influence of the individual components of evolution and the whole, and help us model systems that fall on different parts of this gray spectrum of abiotic to biotic. So now the questions we're interested in this project, and we're interested in answering is, could one dynamic look like another one if we change some things like the fitness function that's used for the selection, or other aspects like the different reaction rates that happen between different reactions, kind of understand the space of possibilities and how complicated the results become in that space, and how could we be fooled? So how could we think that we're actually seeing signatures of one dynamic, but actually it's just another set of dynamic that if you change certain parameters looks exactly the same. So by understanding all of this, we can better understand the space of possibilities. And can we then deconvolute the signals from underlying dynamics? So can we associate certain results with certain dynamics? And ultimately in this project, we hope to understand how complicated this question is and what, if any, set of metrics could help us see similarities between results based on the underlying dynamics. So the goal would be to have something like this where we can kind of like group different results and kind of assign them to different bits of the spectrum. So ultimately, as a community, the goal would be to understand all the differences in dynamics along this spectrum and how they affect the signals we see in populations of polymers, as well as in other chemical populations, because you could expand this work. So I guess I'm nearing the end of my talk already, so we have more time for questions. But I wanted to leave you with this kind of summary that research into the universal features of life can inform measurement requirements. So what do we need from the instruments that we're sending in missions? Because sometimes when you develop a biosignature, you then realize that you need certain capabilities from these instruments. And this research can also guide data analysis and instrument selection. So which instruments would actually be beneficial to being sent on missions, or do we need to actually develop new instruments to make them flight capable? And more general approaches to life detection will significantly reduce the potential for false negative results, which is the key, because we don't want to spend millions of dollars and years of our time to actually go onto Mars or whatever planet and be looking for something so specific that then we miss what's there in the first place that could be something very interesting and that would change astrobiology as we know it. If we came across another life form, it would change everything, because we could actually verify all of these things and verify what's in common between different life forms and our theories of universal principles of life. So I'll end here, and thank you a lot for your attention. Here's a list of all my collaborators, and I especially want to thank Lu Chao, who's co-written a white paper with me and the rest of lab, where a lot of these figures come from that we wrote for the decadal survey earlier last year, and she made a lot of the beautiful figures, so thanks a lot. And to the rest of the laboratory for Agnostic Biosignatures.